Hello, and welcome to Ask an Archaeologist. Ask an Archaeologist is a series of live-streamed video interviews co-hosted by the Archaeological Research Facility and the Phoebe A. Hearst Museum of Anthropology at UC Berkeley. In this series, UC Berkeley archaeologists and others who work with archaeological materials discuss their research and answer audience questions. For those of you joining live today, you may ask a question in the live chat box, which you should see to the right of this video or at the bottom of this video on YouTube. So today we are delighted to have José Luis Marrero Rosado on the show. José Luis is a PhD student at UC Berkeley in anthropology with a focus in archaeology and bioarchaeology. He's currently funded through the NSF Graduate Research Fellowship Program, which will allow him to complete his doctoral research on the effects of structural violence and social identity and differential rates of morbidity and mortality of infectious disease under the supervision of social bioarchaeologist Dr. Sabrina Agarwal. José Luis is also interested in the history of the Caribbean in general, with a particular focus on the historical period. So today he's here to talk with us about what human bones can tell us about epidemics in the past. So welcome, José Luis.
Thank you, Sarah, for that introduction. And I'll start
feel free
right away.
to launch into your slides. If you could tell us a little bit about what is bioarchaeology and your research.
Sounds good. So let me start with sharing the screen. There. So as Sarah mentioned, I am a bioarchaeologist, and the field of archaeology is the study of human remains from archaeological sites, and it's not an old field. It stemmed from physical anthropology, which was more focused on populations, and with bioarchaeology, we focus on what we call the biocultural approach, and that pretty much is how both biology and culture shape human bones and how we can learn about populations in the past by looking at human bones. So bioarchaeology has very broad research interest from evolutionary biology all the way to questions about social anthropology. And it's because of that that it's a very interdisciplinary field. And we integrate things from history, anthropology, social cultural anthropology, and public health, among other fields. With regards to what we do in bioarchaeology with human remains, we usually start by doing what we call a biographical sketch. And this includes the determining the biological sex of the remains, the age, the ancestry, the stature. But then we also look at taphonomy, which is the damage to the bones after burial. We also look at trauma and pathology, and then we also look at diet, migration, and activity in the bones. So now I'm going to talk a bit more about pathology, which is more related to disease. And so paleopathology is the study of ancient diseases. And in archaeology, we include many times paleopathological analysis of the remains. So we are able to do this because many chronic diseases like tuberculosis and syphilis leave evidence on the bone. So tuberculosis and syphilis can be in the humans for months and years. But then you have other acute illnesses like cholera, for example, that are only in individuals for like 1, 2, or 3 days and do not leave evidence. So for those more acute diseases, we sometimes have to focus on biomolecular analysis, and sometimes they help, sometimes we are not as lucky. So here you see an X-ray in this slide of a femur and a tibia from a child. And this was part of my undergraduate thesis. And in this case, I was looking at lead intoxication and the evidence of this on human remains. So individuals that are exposed to high levels of lead, they do not break down the bone normally like they would, but they still build new bone like normal. So then you end up, in this case, I don't know if you see my arrow, these dense metaphyseal bands, and this is just dense bone that is growing but not being broken down. And this is one of the evidence of that this individual was exposed to high levels of lead. Even though this is not an infectious disease, lead intoxication can be an epidemic depending on the social context and where the person lives. For example, Flint, Michigan right now. So this is something that we can look at through bones.
If I could just ask a question about that.
Yes.
What kind of preservation does the bone need to be in, like an archaeological bone, for you to be able to see this kind of damage?
So here, preservation has to be somewhat good because here we are looking in X-rays. So if you have a badly preserved bone, highly porosity, it will be difficult for these dense bands to be visible, especially because when the bone starts to degrade in situ, the edges are some of the first elements that you start to lose. So in this case, we only see these bands in the growth plates, and the growth plates are at both ends of young bones. So for this particular case, they need to be well preserved for us to do that. In other diseases like tuberculosis, for example, you also need good preservation because if the bones are not well preserved, taphonomy, which is the damage to the bone after burial, can mimic some of the things we look at to identify some diseases. So ideally, you'll have a well-preserved bone. That's not always the case. So when you start to lose that outer part of the bone, it can be hard to identify some diseases and illnesses. And in those cases, we might rely on biomolecular tests, including trying to find bacterial DNA in the soil. That's one thing that can be done. Or in calculus, for example, tuberculosis can be identified through analyzing the bacterial DNA in the teeth. So that's some of the things we can do.
Mm-hmm. Thanks.
So, yep. With bioarchaeology and epidemics, archaeology has shown that in many cases, epidemics leads to pretty much the creation of mass graves, and also that epidemics were not that, they did not have such a significant impact on populations until the dawn of agriculture and farming. This is because humans to settle down and populations start to grow. So you have more people living together, and that's what you need for epidemics to spread. Then when epidemics happen, usually you have the creation of these mass graves because there's no space. And then these mass graves usually are left untouched until they're rediscovered. In modern times, it's because usually of construction projects. When they're rediscovered, it's when we archaeologists come in to excavate the remains and analyze the site and see what can learn from what happened there. In archaeology, a lot of the research on epidemics and infectious disease focuses on the Black Death during the 14th century. And the Black Death is caused by the bacteria of Yersinia pestis. And what is important about this is that there were some studies that archaeologists, alongside molecular biologists, that they were able to sequence the bacterial DNA of Yersinia pestis from those historical samples. They were able to determine that the strain was very similar to modern strains. And that suggested that social factors actually played a major role in the spread and mortality of the Black Death, and that it wasn't just a biological factor of the virulence of the strain. So this is very important what we do as archaeologists, because we are aware of that and we look at these assemblages to understand both the biology and the social aspect of these epidemics, because we know that it's not just a biological problem, and that society and culture do play a major role in this. So other things that archaeology touched on is with regards to how groups in power discriminate against other groups, and many times these infectious disease and epidemics have differential rate of mortality, with the poor and the people in lower social class dying at higher rates than those in power. And that legitimizes some of those views by people in power that these people are biologically weak or more prone to disease, when it's just the social, economic, and political aspects that make these people more prone to disease, not just not biology at all. And also in archaeology, we can study how official numbers and reports usually can be biased when reporting death toll, example. And we're able to study that by comparing the demographic of our assemblages to the historical record. And in the Caribbean, I'll mention that in a bit, during the cholera epidemic, there's a big question about that many historians do not trust the official death toll of slaves. And I'll briefly talk about that later when I talk about my project, but this is something that archaeology also studies. And then with archaeology, we can also study how these crises, health crises, alter traditions and behavior in humans, especially as it relates to the treatment of the dead, because this is pretty much what we look at, the evidence of the how the dead were treated. So now I'll
Great.
talk briefly about.
Can I just pause you for one quick sec? If I could just break in, just to remind listeners that they can use the live chat in the YouTube site to type in questions if you have questions for José Luis. Okay, thank you.
Thank you. So with this in mind, I'm going to briefly talk about my own work on cholera in Puerto Rico. So cholera, it's a bacteria, and it has caused over 7 major pandemics. We're still going through the last pandemic, and without treatment, about half of the people that get cholera die. In Puerto Rico, during the 1840s, they were able to keep cholera away by using public measures, but in the mid 1850s, cholera did reach Puerto Rico, and it's estimated that about 5 to 10% of the population died. And these overwhelmed the city, and like I mentioned, usually these epidemics makes cities run out of space to bury the dead. And in this case, the Spanish Crown was pushing for a mass grave outside of the city walls. The church was complaining about all these different things that the government was proposing to treat the dead. And with the epidemic, they decided that they needed to create a mass grave because they did not have the space to bury hundreds of people. So this mass grave was used from the late 1855 to late 1856, and it was supposedly never meant to be again, but they did open it again. And they buried people that died from the yellow fever in 1877. And I'm going to add here what I think is a fun fact of how archaeologists and also historians are able to find this information. So this is actually not something that I found on the archives of the island or the minutes of the Board of Health. This was actually a note that I found on a map from the Spanish fort owned by the National Park Service. Because this mass grave is very close to the fort, so I decided to look at all the maps from that time, and in about 30% of the map, it included the cemetery and the mass grave. And in one of them, it had this note that it was used again. So I mentioned the park, the mass grave is close to the Spanish fort, which is managed by the National Park Service. And recently they extended a trail that used to get just to the fort, but now it's on top of this mass grave, this trail. And bones are being exposed all the time now by people walking there because they did not know that this was there. It's marked. So this led me to create this project. And here you have a picture of one of the maps that I found in the archive so that you have an idea of what San Juan looked like during 1888. This was around the time that the US took over the island. And to the right, you have a modern satellite image of San Juan. See how almost identical it is, even though these two images are over 100 years apart. So that makes my life easier because if they say they name a building, for example, it's really easy to find it today because it's usually still there. So I was able to find the mass grave, and so hopefully we'll start this project next summer. And we'll be excavating the remains and analyzing them. And after the analysis, we plant them in a safer area. And with these remains, I hope to answer my question of how social identity and social violence affect the differential mortality and morbidity of cholera during that time. I want to also look at the demographic of the assemblage to compare to documents. And in this case, what I was mentioning earlier about historians not necessarily trusting the historical documents, it's because during this time, the 1850s, had the abolition movement actually picking up. So then you had the colonial governments in Europe having discussions about the abolition of slavery, but then the Caribbean slaveholders pushing still for slavery because they needed the labor to run their plantations. So a way that they might have done this is to much say that a quantity higher of slaves died from cholera than they actually did, to make this demand that we have no labor here, we need more slaves to keep the plantations going. Mm-hmm. So I hope that also by doing this project, I can answer this of if these records were accurate or not. And then lastly, I also want... Okay, sounds good.
Yeah, do some of these ancient diseases pose a risk of infecting modern archaeologists as they're excavating or studying them?
So, in short, no. And actually, for example, with cholera, we have a big problem on trying to sequence the DNA of the bacteria, even with modern technology. Because it's so degraded that even if we sample the soil and some of parts of the skeletal remains, the DNA is so degraded that we still can't sequence it. So that means that there's nothing alive there. So, but in this case, usually cholera has been buried for 100 years, for example. So after burial, the bacteria is alive there, but then the question is how long. But in short, not too long. So it shouldn't pose any risk.
Great. And so this excavation sounds amazing. And I was wondering, have you started excavating or have you done test probes yet? I mean, what is your work plan? Well, once you can get out there and actually do it.
Yeah, so right now I'm still on the permits part. They asked me for about 6 permits, which is a lot. But I have done some walking surveys and about 3 times different, a few months apart. So I want to do this pretty quick because in every single one of those surveys, I found dozens of human bones on the surface. So it's, and the point is that it's not like complete human femur, for example, that you see it and you say, oh my God, that's a human femur. Because during taphonomy, these are the first bones that are surfacing. So they are easily eroded, and even from people walking, they'll be fragmented. So on another level of the taphonomy is that it's close to niche. So you have also a lot of shells and coral looking things that when people are walking there, if they see this white fragment on the dirt, they'll think it's just like a shell or a coral or even wood, because a bone that has been exposed to the sun, it tends to get lighter in color, and it may look like dry wood, but it isn't.
What do you expect the
So
preservation?
hopefully next summer we'll.
Go ahead.
So that we expect to start excavating next summer if we get all the permits. And with regards to the preservation, that's a great question. We don't, but probably it's going to be. We expect highly fragmented because it's a salty and sandy environment. It might not be the best condition to preserve the bones, but we cannot be certain. So I guess we have to excavate to see how well it's preserved. But from the walking surveys, it doesn't seem like they are greatly preserved.
And how many people, how many individuals do you anticipate might be in this? I mean, I guess you can compare that to the historical record as well.
So the issue with that is that when I started this, I had two questions. Is everyone that died from cholera buried there? And the answer is no. I was able to find some gravestones and pictures of gravestones that say, for example, like this mom and daughter that perished from cholera in 1855, and they have their burial in a private cemetery. And then my other question was, is everyone in that mass grave someone that died from cholera? And also the answer for that is no, because then we know that it was reopened and people that died from yellow fever also were buried there. So then we can just, I guess, estimate what we think might be there, but it could be 10 bodies or it could be 500.
Wow.
Because in San Juan, the death toll was around 500, is one of the estimations, just in the city of San Juan. In cholera and yellow fever, I don't know the specific death toll, but it might be something comparable. So yeah, we have no idea how many bodies.
Wow. And so as I understand, because cholera is a quick disease, you're not going to find obviously any evidence of cholera on these skeletons, but do you expect to find what kinds of other skeletal issues do you expect to find related to disease? And is that going to help you tell like what these people did in their lives and who they were?
Yeah, so that's actually one of the main goals of this, is to study the activity patterns that we can see in the bones, the diet, and other aspects of demographic that can help us understand who was getting cholera and who was dying from cholera. So even though we might not be able to say that someone had cholera from the bones, now that we know that in these mass grave people die from cholera, we can use that data to study like the social aspect of it. And with regards to identifying the cholera, that's another goal that I have, which hopefully I'll be able to do it, and is to find the bacterial DNA, even if it's degraded, to be able to compare to modern and other historical strains. And there is some hope that we can do this with sediment DNA. So instead of analyzing the bacterial DNA from teeth or other parts of the skeletal material, from the actual soil instead.
Right. And so when you talk about the activities people did and that what you can see on their bones, can you give a couple of examples of what kinds of things you can see on bones that would sort of suggest what people did in their lives?
So we look at different things, and most of them is through muscle attachments. So if you are doing a specific movement with, for example, your arms or your legs, those muscles will tend to be bigger, and their attachment will be big in the bone, and the bone will change shape a little bit to try to increase the surface area for the bone for the muscle to attach. So then we can measure those muscle attachments, and we usually do like a level of 1 to 5 in different muscles. And uh, for me, one of the best examples was actually one of the recent PhDs from Sina, and Celise. She looked at cheese making in the medieval period, I believe, and she was able to match some of those activity parts in the arms and other parts of the body to specific movements of making cheese.
Wow.
So that's one of the things we can look at. So in the case of Puerto Rico during that time, something that I might want to look at is what kind of movement do people that cut down sugar cane, for example, and try to match those to some of the activity patterns that are visible in this assemblage.
Great. And this sort of relates to a question coming from the audience, which is, has this kind of work been done at mass graves in other Gulf cities like Puebla and or New Orleans?
So, in terms of research on cholera, I actually have tried to find any other work, and has I have not been that lucky because many times the mass graves are not marked, and sometimes these mass graves are used, in this case, for many other diseases. And in our case, we know that it was only two, so we can probably tell which one is which from the different levels in the mass grave, the stratigraphy. But in the US, for example, it's really difficult to tell because there's no evidence on the bone. So it's not like you can someone say, okay, this one died from cholera, so let's analyze it and see what we can learn. And because we cannot identify the bacteria either, there has probably been analysis of hundreds of remains that of people that died from cholera, but that we don't know they died from cholera. There is a specific project that did look at people that died from cholera in Ohio, and I actually participated in that one, but it wasn't a mass grave. It was a cemetery with individual burials, and in that case, they also do not know if all of them died from cholera. Uh, that's what they think because that was the name of the cemetery. But for that, they're attempting to also identify the bacterial DNA of each specific burial to try to uh, determine which of the remains died from cholera. And in that specific cemetery, I think we excavated about 12 remains, and we identified other 9 burials that needed to be excavated. So it's that will be 20, maybe 30 individuals, which is not as much as what I think we'll be able to find in this mass grave itself one.
And what are your plans for once you, where are you going to do the analysis, and then what happens to all the, say you find hundreds of skeletons, what happens to them after this work?
Yeah, so that's something that we've been wondering too, because if it's, let's say, 80 individuals, we might be able to store them in and somewhere here in Puerto Rico and analyze them here. But then if you are talking about 100 remains, that complicates things a lot because we try to keep each individual in their own containment in order to analyze them later. So if you're talking about 500 remains, you're talking about way too many boxes that you probably won't be able where to store them. So then I think we're waiting to talk with local officials in the city of San Juan and also with the National Park Service to see if they to be part of the project and also provide space for us to store the remains and be able to do the basic bioarchaeological analysis, because for some of those analysis we don't need special equipment, but for the ones that we do, we might take some samples and analyze them. For example, the isotopes lab in Berkeley or the scheduled biology lab also in UC Berkeley.
Mm-hmm.
But we want to keep the remains here as much as possible, and after the project is done, rebury them somewhere safe. And for that, the local community will be involved too. That's also something that we want to pursue, is to have the community involved in this project, because at the end of the day, it's part of our own history here in Puerto Rico, and everyone should have a say on it and take part of it.
Yeah, presumably a lot of these people are people's ancestors, right? Or I mean, it's not that long
because
ago.
Sometimes sometimes like you say like 1855 and think that was so long ago, but it wasn't that long ago.
Yeah.
And you might have many people still living today whose ancestors are there.
Right. So we have one more question from the audience, and I also wanted to invite you, if you had any more slides to show, I'd like you to be able to do that. But okay, great. So this person is asking,
the last one, so.
Perfect. If they're asking, do the bones from the excavation also reflect diet? And could you just comment a little bit about on how you are able to tell what the people ate based on the skeleton?
Yeah. So for diet, it's actually not something we see at the macroscopic level. We actually can comment on diet after we analyze isotopes. We usually analyze isotopes of carbon and nitrogen. And depending on, for example, if the individuals are eating more wheat or more maize, we are going to start seeing a difference on which carbon isotope, the percentages of the carbon isotopes. And then with nitrogen, it's heavily related to meat consumption, because as you keep increasing in the food chain, the heavier nitrogen will be more prevalent than the lower levels. And we can also look at the teeth calculus. And teeth calculus is just dental plaque that gets calcified, and it actually can trap some starch grains, for example, that we usually take a sample of the plaque, we decalcify it and look under the microscope, and we can see some evidence of what these individuals might have been eating. In terms of, for example, malnourishment, we might also look at other indicators of stress, biological stress in the growth of teeth or bone that can also tell us if they were malnourished. So that's one of the things we look at.
So it sounds like you're going to be able to tell so much from these this excavation, and we can't wait to hear more about it. So we're going to have you back in another year or two to talk about what you've been finding there. Thank you so much, yes, José Luis, for joining us today. It was great to hear about your work. And I want to thank our audience, our listeners, also for sending in questions and for joining us. And please join us again for Ask an Archaeologist next week, starting again Tuesday at noon.